Recording in progress.
Okay, it does talk. All right. Fantastic. Yeah, I'm not sure how to mute the Zoom. Anybody knows how to mute the Zoom? My computer's already muted. What?
Probably using this thing as, so you want to go to here, this where you control your audio, select a speaker.
Yeah.
You're doing same system right now.
Yeah, so I'm gonna use this microphone. Yeah. Okay, yeah, this works. This is there.
So what I think I want to do is,
just the speaker.
it's the one, whatever you want to mute.
Yeah,
it needs to be
I think that's it.
Why use like a water bottle? It's brand new, so like no one's ever drinking out of it.
Thank
So
you.
brand new.
Awesome, thank you. Okay, this will be sufficient. Hey, Mom. Oi, mãe. Tá me ouvindo? What? What? I can't? Do you know how to do that? Yeah. I think the beeping stopped now. I think we're good. Yeah, it's good now. It has. Okay, can the people on Zoom hear me? Do this if you can hear me. All right, great. Is the audio good or do you want it louder? Do this if you want it louder. It's good? Okay, great. So sorry, people on Zoom, I cannot hear you. And but this is being recorded, so we'll put it on YouTube or something later. All right.
Is your committee showing up in person?
Matt is on Zoom.
Matt's on Zoom, okay.
Yeah. I don't know if Dave is on Zoom.
David.
Van Valen.
Oh, maybe, yeah, I saw him earlier today. He'll show up. Yeah, we'll give him a few minutes.
Okay.
Yeah, that's all about. Just all about getting the committee. I've drafted people last minute, like someone didn't show up, you just find out it's fax and never get it.
Hey. It's great to see everybody. It's pretty great to be back in person. Pretty happy to.
Can't hear you, Eduardo.
You can't?
No.
What? Can you hear me now?
Oh, Eduardo, you're in person?
If I talk like this, it doesn't knock, or do I need a microphone? I don't know how to set up this microphone.
Hey, Eduardo.
I don't know.
So there's this microphone.
Okay, is there a control panel? Is that Leor? Oh, it's Leor. Okay, so we have everybody, and we're waiting on David, right?
Yeah.
Eduardo.
Where's the thing?
I'm not sure.
No, it should be automatic.
This one.
Yeah, good. I'll try.
Yeah.
Turn that on.
Oh, this works? Yeah, all right. Okay, this is fine.
Yeah, there you go.
Got it.
Give them. We'll assume he's on Zoom.
Okay, that works.
Okay. Me too.
Hello, everybody. We're going to assume that the committee's on here. We got committees on here in three in person, one in Zoom, so we can get started. It's a great pleasure to introduce Eduardo, there's thesis defense. I first met Eduardo driving him and my then 3-year-old son and another grandson, I believe, up to the retreat. And Eduardo, we had this amazing free-range conversation that was interesting to us, not 3-year-old. And it just was a great conversation. A couple years later, Eduardo shows up in my office and says, I'm trying to find a new lab to move to. Here's what I've done. Here's what I'm interested in. And we came up with a plan, which we'll talk about today, as well as the beautiful work he started in Leor's lab. And, you know, just Eduardo, as you all know, will know, is just very talented with a lot of interests, and is very interactive and has a really nice attitude about science and engineering and life in general. And I hope we'll share some of that. And today, as he presents his octopus set of stories about single cell sequencing and interpretation there. So without further ado.
Thanks, Paul, and thanks for everybody for showing up in person and Zoom. I am really happy that I was able to do this in person. The campus is opening up, and we were able to get everybody together. We are going to have a happy hour at Athenaeum at 5, and if you can't make 5, then we make that 6:30 for Leor's Halloween party down in the Zen Garden. So, okay, this is the story of my PhD, and it's not quite just one big thing that I did. It's a bunch of things that I did, primarily driven by curiosity and driven by just the amazing people that you meet being at Caltech, an incredible brain trust that that this place is and what it enables. And these are all projects that were essentially started out of curiosity, you know, and a lot of them kind of started in a whim and they took a life of their own. And I was only able to do so many things because I worked with so many people. Much of this work I did not need. I took a small or a larger part on, but I am, you know, really happy to have done it and to have met all these wonderful people that I will carry on in my heart, you know, as I go on in life after grad school. Okay, so I try to weave together a coherent narrative, but my interests are kind of like far and wide, a little scattered, a little random. I get distracted easily. And today I'm going to tell you about 8 startups, 8 projects that I worked on during my PhD. And the common theme is that they're all about single-cell RNA sequencing. To try to help you keep track of how far we are down in the rabbit hole, I numbered the projects 1 to 8, not in chronological order, but in an order that I thought would help make a bit more sense for someone that is maybe watching this stuff for the first time. And I'll just briefly give you an overview of what we're going to talk about today. I worked on a big database of review of all published single-cell RNA sequencing studies with Valentine Benson. With Sina, we developed an open source range fund for doing experimental single-cell and other kinds of experiments in the lab. Worked on a method for making DNA barcoded beads for single-cell. Helped with Callisto, which is a very fast pseudoliner for processing data. Helped with the CBI tools, which is a fantastic framework for processing single-cell data. Leverage the CBI in multiple projects to quantify how good your data is, to find new marker genes, and most recently to bring single-cell to Wormbase, which is the C. elegans database that Paul coordinates. And so let's dive right on. But before we can go deeply, we're going to take a step back. And as most presentations in biology things we begin with the central dogma. And the 2 things that I want you to take out of this picture, besides the fact that DNA makes RNA makes protein, is that the RNA that's going to make protein, it receives this poly A tail. It is plotted in there. And that's kind of a cheat from nature that lets us capture and look at only that kind of RNA. And then the other important tool that we have at disposal is reverse transcriptase, this enzyme that lets you go from DNA, from RNA back to DNA. And if you're not used to thinking about biology, then think of the RNA as everything that the cell is doing right now, and the DNA as the hard drive. It stores, it's the same in every cell, and the RNA is changing at every moment. And that's why it's interesting to look at it. How do we sequence RNA? And in particular, messenger RNA, which is the interesting RNA that we want to look at. Because it has this feature where it receives this poly A tail, we can readily capture all of mRNA by using a poly-T probe, a poly-T DNA probe, and use it. And then using this incredible enzyme reverse transcriptase, we can convert the sequence that we had in RNA back to DNA. And then we can do a bunch of steps in molecular biology and use DNA sequencing to look at it. And what we get back is a list of each sequence that we saw in each one of the molecules that were present in our sample. Now, why do we care about looking at individual cells? Why do we care about single cell RNA sequencing as opposed to just looking at everybody together at once? And the answer is, it's kind of the difference between you drinking a smoothie and eating each one of the individual berries. You know, if you have a lot of different berries, then when you blend them all together, you get an average of how they taste like. And if there are very few, you're not going to taste a lot of the blueberry flavor. And with RNA sequencing, it's the same thing. If you have a tissue, you know, a sample that has many different kinds of cells, and you blend them all together, you're only going to see the average. And if they are very different amongst themselves, you're going to mask all the interesting stuff that's happening there. Everything that makes biology work. You have the brain, which has hundreds of different types of neurons, of cells. You're going to lose all of that if you're just doing a smoothie. On the other hand, if you're able to purify your sample, or you have a tissue that has all the same cell types, then maybe you don't need to do single cell. If you're a smoothie, if you're just drinking orange juice, maybe it's fine. You don't need to taste each one of the berries individually. But the truth is that it's really, really powerful to do this. And a testament to this power is how fast it became widely adopted, this single-cell RNA sequencing techniques. Over the past 10 years, if you look on the y-axis there has been essentially a million-fold increase in the number of individual cells that we are able to profile in a study. 10 years ago, it was a few. Now we can easily do a million. But if you look at this graph here, which ends, it ends in 2017. And this was from a very popular review by Valentine Benson, who had then finished his PhD, and he came to Caltech. And he came to work in neuroscience. And we ended up hanging out a lot. And one of the things that we did together, out of a Google spreadsheet that Valentine had, where he was collecting the different studies that he saw every now and again for his own personal use, was we converted that into a comprehensive database and as a resource to the community. And what that lets you do is you can go back there anytime, because we continue to update it. And you can see the current state of the queue. And it just kept growing and growing and growing. And I think now it's going to become more and more of a routine technique to do single-cell RNA sequencing almost every time, because you're almost always dealing with tissues that have many different cells, and you do benefit from being able to tell them apart when you're doing your study or research. And right now we have over 1,200 published studies that are tracked in this database, and it's very nice to just follow how the field has been growing. But you're probably asking, how do you do 1 million cells at once? Or how do you do 1,000 cells at once? And there's basically 3 different ways that you can do it. In the beginning, people would isolate individual cells in physical compartments and putting the DNA of the poly-T capture sequence in a barcode that would identify where the cell came from. But that's it's kind of hard to scale because we've got a lot of plates. Then people came up with these techniques that are split and pool. So if you can sequentially grow your barcode, you don't need to barcode all of them at once. So what you do is, if you have 3 different colors that you can paint your cells, you split them in 3 groups, paint them each one a different color, pull them back together, mix them well, split them again, and do that 2 or 3 more times to grow a barcode. And then combinatorially, a cell is going to almost certainly go through a unique path there, and they will receive a unique barcode. And that's one of the ways that it really can scale to millions of cells in a single experiment. But probably the most, or certainly the most popular type of method are droplet-based methods, where you do isolate the cells in physical compartments, but it's not a well, it's droplets. And this droplets is generated in a microfluidic device that is pinching it with an oil flow. And together with the cell, if you can see on my screen, there comes a DNA-coded bead. And that bead is what contains the poly-C sequence and the barcode that will allow you to, when you do your sequencing, identify from which cell each read came from. And what you see here at the bottom, this video, shows you a bead coming in, this big black, and a cell coming in here at the bottom, this little thing. A bead is about 60 microns, and a cell is usually like 2 to 10, depending on what kind of cell. And so there were many different types of droplet-based methods. And to run them, you use this microfluidic strips to make these emulsions. And so one of the first things that we were doing at the Pacto lab is we were trying to run this emulsion and microfluidics generating chips. And one of the things that you use to run these things are syringe pumps to control very precisely the flow rates. And there was a bunch of 3D printed pieces of a pump, half assembled, lying around the lab that Jay Scaring, who was then a grad student with Leo, was trying to assemble. And I came in and I saw that and I said, you want to 3D print some pumps? I think I can help. And I think I know a guy that can help. And so I went to Sinan and I was like, hey, Sinan, do you wanna do some 3D printing and CAD design on the side? And Sinan was like, yeah, sure, why not? And that side project became a big project, because what we did here is we started from this published pump design and this other microscope station design, and we kept iterating and iterating and iterating until we arrived at a design that was like super easy to print, could be obtained with parts from Amazon, nothing specialized, and would actually allow you to run this microfluidic chips. And single cell was the motivation to do that. But in the end of the day, what happened was that work taught us a lot of lessons about how to build a piece of lab instrumentation. And together with the pumps and the designs that are available on GitHub, we published these guidelines on things that you might want to keep in mind if you decide to build up your own 3D printable open source piece of lab instrumentation. And today this pump has been used by a lot of people. Every now and again, we hear of someone that's making some modification. And we are already happy with how it turned out. And I think it is the attention to caring about documenting your project well, making it simple, making it really well tested, robust, making it so that you can mix and match the different pieces, making it modular, and benchmarking it, showing people what they can do with the project. These are some of the principles that we included from our own experience and that we shared in this work. And so I said that there are many different kinds of beads. In fact, our next story is about how also at the Pacto Lab, we sought to improve on how you make one particular kind of bead that is a hydrogel based bead. So Alan Klein and others have published this method called pin drops. teaching you how to make these beads, how to operate these microfluidic chips that you saw with the pump. And they had made a really cool protocol for producing these DNA-coded beads. And we saw that and we thought, maybe we can tweak a few things and make it a little more flexible, a little more easy to produce. And so the entity that we ended up improving upon is essentially this that you see here. It's a hydrogel bead, and it has to have a DNA barcode coding it that is unique to each bead, and it has a structure that's it's kind of like this. You have a barcode that's comprised of different parts, and you have a branch sequence that's called a UMI, and you have a capture sequence that is just your poly-T that you use to capture the mRNA. And to make this protocol better, the main thing that we changed is we used this really cool reaction called PER, primary exchange reaction. And this reaction is really cool because DNA acts as a catalyst. It is not consumed in the reaction. And so what you see here is you start with a piece of DNA that you want to extend, and in comes a hairpin. And what you do is your hairpin only has 3 letters, let's say A, T, and C, and it doesn't have G's in solution. And it doesn't have G's in the hairpin except in the spot. And so it's going to anneal to your DNA, and it's going to look like this. And then DNA polymerase can come around, can start elongating and displacing this strand. And then eventually it's going to get to the G, and it's going to stall because it can't go further because there's no more G in solution. And so it falls off. And when it falls off, there's this process called strand displacement, or that makes the hairpin compete with the newly synthesized strand, and then it anneals back. And eventually, because there's only a few base pairs, boom, it falls off. It can come back here because of this new pink region is blocking it. And it's free to go and catalyze another reaction. We also did some other improvements on the gel chemistry, and that work is now being carried forward by Tali, who is another graduate student with Lior, but it is also described in a provisional patent that we filed a few years ago. So that was a very, very interesting improvement and fun method to develop. And that was my main experimental project during the PhD. And so now we're going to kind of switch gears and go more into the computational and analysis realm, because I'm not really good at the wet lab. I kind of do it like in cooking, and it's not really good practice. It's not the ideal. I really like doing computational stuff more. And so now we transition to our computational stories, and the thing that you want to do after you get your data back from the sequencer is, okay, you've got a huge FASTQ file. There's a bunch of lists of sequences, and you want to align it. You want to count how many molecules of each kind you saw, and so that you can get this object that's called the gene count matrix. And then you want to tell apart which cells correspond to good cells, which cells might be just empty droplets, so that you end up with a filtered gene count matrix. And so this next story that I'm going to tell you is about the first part of this process of realignment or pseudoalignment. And what this task consists of is you get your FASTQ file, which looks kind of like this. It's just a bunch of sequences. And each sequence has a specific structure. The cell barcode is here. This identifies from which side it came from. And you have the UMI, which is just a random sequence. The point of the UMI is that if you get 2 copies of the same original molecule, they will have the same UMI, so you can tell that they came from the same molecule, so you count it once. Then you're going to have another region that is the cDNA, is the part that corresponds to the transcript that you copied from your RNA template. And so essentially the task that you have here is we've got to group this by which cell they came from, which barcode. We got to deduplicate the MI, so make sure it only counts each one once. And then you identify from which gene they came from, and we count how many copies of each gene you saw, and you put all that in a matrix. And that matrix is the gene count matrix, which is the fundamental object of single-cell RNA sequencing, where the y-axis you have genes, and the x-axis you have cells, or flipped. One axis you have cells, one axis you have genes, as opposed to just one big list of how many copies of each gene you saw in the sample. Now you have many samples because each cell is a sample. And the project that I worked on with Leo to make this better was the Callisto bus package, which Callisto was a software package developed by Leo and colleagues back in 2016 to make super fast quantification of bulk RNA sequencing. But bulk RNA sequencing and single-cell RNA sequencing are essentially the same beast, except that you have this thing with the UMI and the barcodes. So it was a relatively small change that would enable you to use the same process of pseudo-alignment, which is exact string matching, to count the number of transcripts in a single cell set. And the really, really cool thing about Callisto is that it's extremely fast. It's extremely memory efficient. And it works, the results are essentially the same that you get when you're using a real line like STAR or Cell Ranger. I helped benchmark and show that they were indeed very, very similar. It's so fast, in fact, that back when we benchmarked it, it was about 30 to 40 times faster than Cell Ranger, which means that instead of taking 30 hours to do a run, you could do it in 1 hour, and it will take 5 times less RAM. So instead of taking 60 gigs, it will take 8. And so that opens the door for doing this on your desktop or reprocessing all data sets again and again as the references change or as you want to do something different. And like we did with the Poseidon pumps, we made all of this well documented and available in the Callisto bus website that you see there. And this work came out recently in a few months ago. And so we got the gene count matrix. We used Callisto. It was really fast. Now what do we do with it? There is a lot of different methods that people use to work with single cell data because matrices are the fundamental object of linear algebra. They have been around for a while. And so I'm not going to attempt to give you a comprehensive landscape. I'm just going to zoom into one thing that I think is really cool and which I worked on, which is based on the idea of having generative models or Bayesian models for your data. And the idea of generative models is instead of having to compute a specific formula for any statistic that you want to compute, if you're able to learn a distribution to generate your data, learn a posterior distribution, then you can just sample from that posterior, and then you can compute any kind of statistic that you're interested in. And this turns out to be really, really powerful because it amounts to just a lot of computation, but all the methods become relatively the same when you want to when you want to adapt them to different domains, when you want to adapt them to different quantities that you are calculating. And what happens with a generative model is that you have your original data. You are going to learn a latent representation. Each point is going to be parameterized with a given set of numbers, and you're going to be able to, for that cell type, let's say that I parameterize the green cell type, I'm going to be able to generate new cells that look like the green cell. And how good the new cells that you generate look like the original cell is the metric that you try to optimize when you're learning these distributions. And it enables modeling in a very flexible manner. And this is made particularly easy by this package that's called SCBI tools, which I also worked on, mostly documentation, that takes away all of the plumbing, all of the nitty-gritty aspects of just coding, putting together your machine learning framework, and lets you really focus on the model that you're building for the application at hand. And so SCBI currently has many different data modalities that it can that you can work with, and it has within each modality you even have different models depending on how which phenomena you wish to account for. I'm not going to go into the specifics because it will take quite a while. But basically, it's a modular way of specifying what models you want to build, how you want to train them, and allowing you to compute all of these statistics in a very streamlined manner.
Eduardo, so by model, an example of a model would be that these data are generated from real cell types. And you're trying to explain how you're getting the variation at the single cell level from those populations of cell types. Is that the model? Can you go back a slide?
I have a so this is an example of a model, which I mean to say, the process of generating data for single-cell RNA-seq is essentially the process of sampling from an arm of balls. And the way that you parameterize how you sample from an arm of balls is with a multinomial distribution. Now, you might do something like first you sample from the arm of balls, and then you toss a coin to see if you get a 0, or if you do see the sample that you just draw. So that will be a two-step model. And what SCBI tools lets you do is it lets you build this conditional hierarchical models in a very straightforward manner, where you can concatenate these distributions in such a way that you can account for different steps of what is happening, and then you can train it without really having to worry about the plumbing. And so, as long as you're able to describe the different steps in which the process of the model, then SCBI takes the rest away. Yeah. Sorry, I thought that slide would be a little too much, which I kind of had skipped it. So using this idea of how good is the data that you're generating, how close is it, to the real data? Using this idea of the reconstruction error, it offers you a way to quantify. It offers you a metric for the goodness of your data. Presumably, if your data is more informative, we're going to be able to learn distributions that can generate cells that look more like real cells. And so we leverage that to quantify the goodness of different kinds of single cell data sets. And so in machine learning, people typically talk about, oh, let's try many models in one data set and see which model performs the best. But with Valentine, we flipped this problem on its head, and using SCBI, we asked, let's try many data sets on the same model and see which data set works the best. And we were only able to do that because we had SCBI and we had Callisto to enable us to reprocess uniformly the same data sets over and over and over again. And so what we did to do that, the idea was we would take the full FASTQ file, we would subsample it to simulate if you had done less sequencing. And then you would run Callisto, you would get the count matrix, and you would subsample this count matrix, and you would train CI on this different count matrix. So you have two axes. You have subsampling on reads, and you have subsampling on cells. And you would run the CI, see the error, the training error, and then make a plot to kind of like get a sense, a t-SNE plot, to get a sense of how good was the reconstruction of the data, like how much structure could you see in the data. And our thought process was, if we use this training error as a metric of like the goodness of the data, then we could say at one point it will be better to either do more reads or to do more cells. To illustrate that, we created this grid plots, which might be a little overwhelming, but in one axis, on the y-axis, you have the number of cells that were sampled, and then on the x-axis, you have the total number of reads. And on the top plots, each dot corresponds to one of the points, one of the subsample data sets. And dark blue is lower error, so it's better. And yellow is like worse. And one very popular thing that people like to do in single cell is they like to make these t-SNE plots that kind of let you see if things are like clustering together, if they're like different, or if they're all the same. And what we saw was that with the data sets that had like very few cells or very few reads, it's basically just a blob, a single blob. It had no structure, which means that the model wasn't learning anything because the data wasn't very informative. And as you added more reads, as you add more data, or as you add more cells, this kind of substructures begin to appear in the data. So you get more blobs. And this is just a very quantitative way of looking at what's qualitative way of looking at what the top plots, which is more quantitative studying. But as you get more reads and more cells, of course, your data is better to train this on. You use this quantification for this data sets, we're able to find a threshold for only a few data sets that we look at, where beyond 5,000 UMIs, it was better to just do more cells, not do more reads. So we're talking about visualization, and that's a really, really big part of data analysis of any kind. You need to be able to look at and get a sense of the data that you're working with. And we need better tools to visualize it, to get a sense of what we're doing. It's not enough to just run the black box and hope that it works, because each data set is different, and often there are nuances. And most importantly, we need these tools to be user-friendly. If we need to write a long piece of code, even a person that's good at writing code will not be able to do a lot of analysis. And when you need to be handing it off on and off between different people, that also hugely hinders the speed of interaction between wet lab and dry lab. And so my work with Leo really motivated me to think about how can we make better tools to apply to single cell biology that do the tasks that people want to do in a way that's straightforward and that's useful. And so the last two stories that I'm going to tell you about are about data visualization and what that enables. And so this first, well, story number 7 is about an idea that Mark asked me last year. Mark asked me, like, how would you find marker genes for a given cell type out of single cell data? And I said, well, I think that a marker gene is a gene that is high expression and specific to one tissue. So if I were to look for them, I would first quantify how unique it is to only that cell type, quantify its expression, and then visualize its expression across all other cell types in the organism in the data set of interest. And to visualize that, I came up with this swarm plots that I call them, which all that you're seeing here is on the y-axis you have a list of genes, and then on the x-axis for a given gene, what you have is you have a reference cell type. So in this case, you have ASG as a reference cell type, and you see the expression of in every other cell type relative to ASG in terms of logical chain. It's missing the Y here, and which means that everybody to the left of the 0 has lower expression. And if you want to find a cell type that only has that gene there and nowhere else, you just look for which ones only have dots to the left of the dashed line. And then if you want a really good marker, then you see, well, okay, I got a few candidates who has the highest expression. And so very simple idea. This quantification and everything was done using SCBI. And I made a plot very similar to this because they asked me, well, could you help us find candidate marker gene for warm neuron ASG? And I said, okay. And I made a very similar plot to this. And I looked at one of the top hits that had very high expression. And the first gene, Y41C4A6. And I said, this looks like something you should try. And they said, okay. And didn't hear anything for like 2 months. And then one day I got an email that said something along the lines, oh my God, I never saw a neuron so bright. Look at these pictures. And I was like, whoa, science works. What you're looking at here is a warm neuron that is brightly labeled by this gene Y41C4A6 that we found by making this ad hoc plots with public single cell data. And since then we found a bunch more. I didn't do any of the experiment work. That's all the wonderful members of the Stanford lab. And this workflow is described in this GitHub repo. The manuscript is still in preparation, but we've had about 50% success rates, which is pretty remarkable. It speaks, I think, primarily to the quality of the data that we're using, and not so much to how clever this was. So I showed you one example of, if you make a tool, a visualization tailored to a specific task, you can get a lot out of it, especially if you make it like interactive and you let people explore the data on their own, but with features that are actually useful. And so the primary thing that I've done during my time at Sternberg Lab was trying to bring these tools and make them more useful for a broader audience. And so in work that I did with Valerio, who's sitting there on the back, raise your hand, we developed the single cell tools for worm-based, which are a suite of interactive tools that are deployed for almost all the single cell C. elegans data sets out there, which is, say, three studies. And they let you do differential expression, they let you do this swarm plots that I just explained, visualization of the expression with histograms and heat maps in a way that gives the control to the user, to the biologist that wants to find the answers and doesn't have to rely on a bioinformatician being there and taking a week to get back with the results, and then it's like the wrong thing, and then you send out over like another Excel sheet. This takes knowing like both words. This takes knowing like what are the needs of the biologists and what are the methods that you would want them to use if you could suggest which way to analyze the data. And so we are very happy that these tools are now deployed at worm-based. They are also available on GitHub. If you just go to single-cell.wormbased.org, you will find it. You'll find the GitHub repos. They're super easy to deploy. You can deploy them to your own data sets. You don't need to only work with C. elegans. And this paper, it's on the bioRxiv and is now under revision. So hopefully gets accepted in the coming months. And so that's the last story that I want to tell you about. I just wanted to close with just a few thoughts, which I think are not so much about the papers and the publications. That's the part that I focus on this presentation. But what the thoughts that I want to close with is about people. It's like all of this was only possible because of everybody that I met at Caltech that enables you to do this kind of curiosity-driven science that enables you to work with amazing people and do things that really can't be done in other places because you can't bring two totally different disciplines together. Today's kind of like a bittersweet for me because I wish I could be here a little longer, but life happened, and I'll tell you after the acknowledgement slides. I just want to say thank you to everybody. And I guess that now I do my acknowledgements. And so, first of all, to all of my friends and collaborators and everybody that has ever helped me on troubleshoot something. It really makes everything so much more fun. But out of everybody, I want to zoom in into a couple people and call them out. Paul, just being like incredible and giving you like all the intellectual freedom that you could wish for to do whatever you want, and just give you the trust that that you're going to finish the work, get it done. Valerio for working with me on the basic single cell tools and being awesome, reviewing all my crappy code. Mark for somehow turning what was just a hunch into a brilliant experimental project and leading that really. I only did a couple plots. It was really awesome working with you guys. And then before that, the people from the Lear's lab, Lear, of course, Aline for taking up the BID project and carrying that forward. It's Karen for teaching me most of what I learned about wet lab. Sina for a lot of late night discussions, a lot of work done together. It's always been fun, 3D printing and designing stuff. And then a few other people. Well, Valentine Benson from Fractal Lab, who's now in Boston. Certainly, we spend a lot of time together, and Valentine was probably the single person that taught me the most about single cell, and always willing to answer my questions somehow. And I had a really good time hanging out with Valentine and learning a lot from him. And I'm just really happy that we crossed paths at Lab for a couple of years. This next one is sitting right here wearing the Oklahoma hat. Dylan, thanks for being my friend and being always willing to embark on all of the craziest possible side projects that one could conceive. Like, I think the only thing that kind of panned out was the pumps. But like, yeah, if I were to tell you all the stories of all the random stuff we tried to do together, it would be another hour. And Dylan was always down to try whatever weird stuff. That's why half of the pictures he just eyeing me suspiciously, because he's like, what do you want to do now? Like, why? So thanks to Dylan, and also for always putting up with my bad jokes. Thanks to David, Tatiana, and Jeff, who are now in Silicon Valley doing their startup from Thompson Lab. And this center picture here was taken at the Lone Incubator, where they're now doing their own company. And I'm really happy that we are going to be able to hang out together more in the future, because we're all going to be in the Bay Area. Caltech is not super famous for its vibrant student social life, but it's there. If you search for it, it really is there. To all the happy hours, all the social events, all the things that really bring the community together, and let people just hang out and exchange ideas. In particular, to the CMS happy hour, which is hosted at the Athenaeum every Friday until the pandemic hits, which is this picture here, first picture. Certainly my favorite happy hour at Caltech, and I do hope that they resume as soon as possible. Neuroattackers who bring together, the students that have some kind of interest in neuroscience to hang out. The Caltech Y, which does incredible work, giving students other opportunities to explore different topics, to explore policy, governments, things beyond science. South Club, which is my politics club, which is now every Wednesday, used to be in person, and now it is on cyberspace on this very Zoom link that was sent out to the presentation So if you come to the Zoom link Wednesday 6 p.m., you're going to run into us talking mostly about politics and having a good time. Thanks to my mother, Angela, and to my sister, Bea, for raising me and just letting me do whatever I wanted. Kind of worked out. Thanks to Retro Biosciences, which some of you may know, I'm joining full time now that I'm graduating. And is the crazy biotech startup in the Bay Area that I'm going to be working at going forward. If any of you are ever around the Bay Area, just let me know and let's hang out. Finally, a little tangent, not about science. Thank you to the Wilson Grow Institute in my hometown of Florianópolis in Brazil. This past year afforded me the opportunity to go back there and work remotely and take a step back and think through about what is important, what are the things you care about, what are your values, what are the things you want to do, what are the things you want to affect, and how could you possibly do that? And through a year of reflection and thinking, what could I, in my capacity as grad student, do? I found this wonderful group of people who have 40 years now been doing outstanding social and educational work in marginalized communities in Brazil, and affording them the opportunities that I've had, which are the reason that I'm here today, because I got an exchange program. I got to go to the US, and I was just afforded these opportunities. And so, going forward, I am really happy to be able to support them strongly. And if you guys are interested in that kind of stuff, or you're thinking about what's important to you, I highly encourage you to take your time, take a step back, think about what are your values beyond just your research. What are the things that you wish to see in the world? What is important, right? Beyond just the next paper, like what's the broader picture? Because you're going to be around for a while. So what is the direction that you want to make a push in? And so that's it. If you want the occasional updates and next chapters of these stories, that's my website. It's not updated very frequently, but you can check it out on occasion. Thanks.
We'll have questions from the audience. Questions?
All right. Throw the microphone.
And on Zoom, maybe Zoom.
Oh, yeah.
Hello?
I have one question. So your depth versus number of cells chart,
Yes.
you sort of topped out at 10,000 cells.
So it popped out at, for those data sets, it was 5,000 UMIs, which corresponds to about 15,000 reads per cell for the 3 data sets that we looked at. But it can be different for other data sets.
But I think it seems like you were going above 10,000 cells, that's probably a flow cell, right? So what if you went to 100,000 cells or 1 million cells? When you start seeing the blobs get, 2 million blobs?
Yes, you would if you did more cells, more and more cells with sufficient depth, you would see more and more subblobs. So subpopulations would cluster together if you do 1 million cells, and you had the heterogeneity in that sample. So if you're doing a brain sample, they would see all the subtypes. But if you're doing like kidney, or liver, which are hepatocytes, then it would be mostly just a few blobs, even if you did 1 million cells.
Okay, so you're not getting artifactual blobization.
You could be. The blobs were just a fun way of visualizing it. The quantification was SCBI.
Question. Please, Mike.
Hey, good job, Eduardo.
I got a question about at the very beginning where you talked about the splint pool and dropsy. So it seems like the splint pool, you can scale it a lot better.
Correct.
But why did you guys commit yourself so heavily toward the droplet approach?
That was basically because at the time, those split pool methods were not super popular. These droplet-based methods were like all the rage. And this protocol for making the beads was really good. And most importantly, if you're making your own beads, you can put a sequence on the bead. That's not just a poly A. And so the real prospect that we had there was to capture things that were not just mRNA, was to capture other kinds of RNA in the cell, like long no-coding RNA. Those things we never look at with single cell. So doing beads that had a custom capture sequence, that was the most appealing thing to do with that project.
No further questions. Let's thank Eduardo for a really beautiful presentation.
No, can you hear me? I think.
And we use the thesis next year.
I guess, or we can go somewhere else. I don't know what's the.
Okay, so thank you everybody for coming, and we're gonna retire. You're gonna retire. We're gonna stay here for the closed session.
All right. Thank you everybody that came on the Zoom. I'm going to close the meeting now. Matt's there.